Continuation of the previous video, cephalosporins. Earlier we mentioned about penicillins. Next, cephalosporins. Cephalosporins originally isolated from the fungus Cephalosporium, and their beta-lactam structure is very similar to that of the penicillins. So, members of the cephalosporin family of antibiotics are effective in preventing the synthesis of bacterial cell walls and mostly useful against gram-positive bacteria. Then tetracyclines. The tetracyclines are a family of antibiotics with a common four-ring structure to which a variety of side chains are attached. Tetracyclines are broad-spectrum antibiotics effective against gram-negative bacteria, gram-positive bacteria, Rickettsia, Chlamydia, and Mycoplasmas. Then aminoglycoside antibiotics. The aminoglycoside antibiotics are inhibitors of protein synthesis in bacteria. The aminoglycoside antibiotics are inhibitors of protein synthesis in bacteria. Members of this family of antibiotics all have amino groups and carbohydrate molecules associated with the main molecules. The members include gentamicin, kanamycin, and then tobramycin, streptomycin. The aminoglycosides are most active against gram-negative pathogens. Then erythromycin is a macrolide antibiotic is synthesized by Streptomyces erythraceae. The macrolides contain a 12 to 22 carbon lactone ring linked to one or more sugars. Then erythromycin is useful against gram-positive bacteria and acts as inhibitor of protein synthesis. Then chloramphenicol. Chloramphenicol was produced from cultures of Streptomyces phenazulae. It is now made through chemical synthesis. Antibiotic has a very broad spectrum of activity. Mechanism of action, target of some antibiotics. Antibiotics: penicillin, vancomycin, bacitracin. Inhibit transpeptidation enzymes involved in the cross-linking, binds directly to the DALA terminus and inhibits transpeptidation, inhibits cell wall synthesis. Viruses. Next is about viruses. Viruses are simple non-cellular entities consisting of one or more molecules of either DNA or RNA enclosed in a coat of protein. So, I am interested in developing an AIDS vaccine in the future. So, I am quite interested in this. So, next is about virus. I'm to a great extent interested in viruses because I want to invent a vaccine for AIDS. I hope it will happen. Viruses are simple, non-cellular entities consisting of one or more molecules of either DNA or RNA enclosed in a coat of protein. They can reproduce only within living cells and are obligate intracellular parasites. Viruses are smaller than prokaryotic cells, ranging in size from 0.02 to 0.3 nm. Smallpox virus is largest virus, about 20 nm in diameter, and poliovirus is the simplest virus, about 28 nm in diameter. A fully assembled infectious virus is called a virion. The main function of the virion is to deliver its DNA or RNA genome into the host cell so that the genome can be expressed, transcribed, and translated by the host cell. Each viral species has a very limited host range. It can reproduce in only a small group of closely related species. Viral structure. The structure of virions are very diverse, varying widely in size, shape, and chemical composition. All viruses have a nucleocapsid composed of nucleic acid surrounded by a protein capsid. A protein coat the capsid, which functions as a shell to protect the viral genome from nucleases and which during infection attaches the virion to specific receptors exposed on the prospective host cell. Capsids are formed as single or double protein shells and consist of only one or few structural protein species. The proteins used to build the capsid are called protomers. The nucleic acid associated protein called nucleoprotein, together with the genome forms the nucleocapsid. Some viruses have a membranous envelope that lies outside the nucleocapsid. Those virions having an envelope are called enveloped viruses, whereas whereas those lacking an envelope are called naked viruses. In enveloped viruses, the nucleocapsid is surrounded by a lipid bilayer and glycoprotein derived from the modified host cell membrane. So enveloped viruses often exhibit a fringe of glycoprotein spikes or knobs, also called peplomers. In viruses that acquire their envelope by budding through the plasma membrane or another intracellular cell membrane, the lipid composition of the viral envelope closely reflects that of the particular host membrane. Viral genome are smaller in size. The largest non-viral genome last of bacteriophage gene is 670 kilobase pairs. The genome of a virus may consist of DNA or RNA, which may be single-stranded or double-stranded, linear or circular. The entire genome may occupy either one nucleic acid molecule, monopartite genome, or several nucleic acid segments, multipartite genome. The genomic RNA strand of single-stranded RNA virus is called a sense, positive sense, pulse sense in orientation if it can serve as mRNA, and antisense, negative sense, minus sense if a complementary strand synthesized by viral RNA transcriptase serves as mRNA. Then shape symmetry. All viruses have a nucleocapsid, nucleic acid and protein structure. The symmetry refers to the way in which the capsomers are arranged in a virus capsid, may be icosahedral, spherical shape, and helical, rod shape. Helical symmetry is seen in nucleocapsids of many filamentous and pleomorphic viruses. Helical nucleocapsids consist of a helical array of capsid proteins, protomers, wrapped around a helical filament of nucleic acid. A typical virus with helical symmetry is TMV. An icosahedral morphology is characteristic of the nucleocapsid of many spherical viruses. An icosahedron is a regular polyhedron with 20 equilateral triangular faces and 12 vertices. Complex structures have capsid symmetry that is either purely icosahedron or helical, as for example T4 virus of E. coli. In classification of viruses are classified on the basis of their nucleic acids characteristics, capsid symmetry, and presence or absence of an envelope, their host, dimensions of the virion and capsid, and other properties. Some common virus groups and their characteristics. The International Committee of Taxonomy of Virus, ICTV, has described almost 2,000 virus species and placed them in 3 orders, 73 families, 9 subfamilies, 287 genera. Virus order names end in virus. The International Committee for Taxonomy of Viruses has described almost 200 virus species. Viridae, Virinae, and virus. The Baltimore system of virus classification is given: double-stranded DNA genome, single-stranded DNA genome, double-stranded DNA genome, group 1 to 7. Baltimore's simplified classification of viruses. This classification is based on the type of nucleic acid in the genome and the mode of production of mRNA. Bacteriophage. Bacterial virus. Phages were first observed in 1915 by F. Trott in England and in 1917 by F. de Herelle in France. Named them bacteriophage. Eaters of bacteria. Several morphological distinct types of phage have been described, including polyhedral, filamentous, and complex. Complex phages have polyhedral heads to which tails and sometimes other appendages, tail plates, tail fiber, etc. are attached. Capsid head, nucleic acid, collar, sheath, base plate, tail fibers, tail pins. T-even coliphages. Single-stranded DNA phage, double-stranded DNA phage, single-stranded RNA genome of plus sense, double-stranded RNA genome. Life cycle of a bacteriophage. For a virus to replicate, it must induce a living host cell to synthesize all the essential components needed to make more virions. These components must then be assembled into new virions and escape from the cell in order to infect other cells. There are two different types of viral life cycles, lytic and lysogenic cycle. Lytic or vegetative life cycle culminate in the lysis rupture of the host cell and the release of numerous viral progeny. Bacterial viruses exhibiting a lytic life cycle only are also known as virulent bacteriophages or lytic phages because they inevitably cause the death and destruction of the host bacteria. Examples of virulent phages are the T-even phages. The lytic life cycle consists of 5 steps. There are attachment, penetration, synthesis, assembly, and release. The cycle begins by the attachment of the bacteriophage to specific host cell molecules or receptors on the bacteria cell wall. After the virus attaches to its host, it introduces its genetic material into the cell. Penetration. Once the bacteriophage genome enters the cytoplasm, it subverts the host's nucleic acid and protein synthesis appears and initiates the synthesis of viral proteins and DNA. As the virus proteins are synthesized, they self-assemble into viral components such as the head containing the phage DNA, tail and tail fibers. The assembly process results in the formation of numerous intact phage reticula within the cell. After the assembly step is completed, viral proteins cause the lysis of the host cell and all the viral progeny are released into the environment. The average number of phage particles produced by each infected cell, called the burst size, is characteristic for virus and often ranges between 50 and several hundred. Virion DNA cell host protein coat remains outside, viral DNA enters. Attachment, adsorption, penetration or injection, synthesis of nucleic acid and protein, assembly and packaging, virions release, lysis phase. The lytic cycle of a bacterial virus. Because the release of virions is a more or less simultaneous process, the virions are said to undergo a one-step growth curve. One-step growth curve. Relative virus count, time, latent period, eclipse maturation. The one-step growth curve of virus replication. This graph displays the result of a single round of viral multiplication in a population to cells. Following adsorption, the infectivity of the virus particles disappears, a phenomenon called eclipse. This is due to the uncoating of the virus particles. During the latent period, replication of viral nucleic acid and protein occurs. Then lysogenic cycle. In the lysogenic cycle, the viral DNA is inserted into the host DNA and reproduces as the host DNA replicates. A lysogenic virus, temperate phage, can remain in this stage for numerous replications of host cell DNA until it exits itself from the host DNA and undergoes a lytic life cycle. When the genetic material of these phages is inserted into the DNA of host cells, it is said to be in a prophage stage. A cell that contains a prophage is known as a lysogen. The physical state of the prophage is not identical for all temperate viruses. For example, the prophage of the bacteriophage lambda in E. coli is inserted into the bacterial chromosome at a specific site and replicates as part of the bacterial chromosome, whereas the prophage of bacteriophage P1 in E. coli replicates as an extrachromosomal plasmid. An example of a lysogenic virus is the bacteriophage of E. coli called lambda. The lysogenic bacteriophage lambda, like phage T4, also uses its tail to inject the viral DNA into the host. Once the viral DNA is inside the host, it can either undergo a lytic cycle similar to that of T4 or incorporate itself into the host DNA. Injection of linear phage DNA, circularization of phage DNA, integration of prophage, lysogenize cell. The integration of the circular lambda genome involves the presence of an attachment site in the genome of both bacteriophage and bacterium. The bacterial attachment site, called ATTB, consists of a specific DNA sequence composed of 3 domains, a B domain, an O domain, and a B' domain. The attachment site of the bacteriophage, called ATTP, is also made up of 3 domains, a P domain, a O domain, and a P' domain. The O domain has a sequence common to both ATTB and ATTP. The O domain is also known as the code sequence. Lambda DNA, POP-BOB, ATT sites, bacterial DNA, integration of the bacteriophage lambda genome into E. coli chromosomal DNA. Lysogeny is maintained by a repressor protein encoded by the DNA of the lambda prophage. The lambda repressor blocks the expression of the other lambda genes, including those responsible for phage replication and synthesis of phage proteins. An intriguing property of lysogene is that they cannot be infected by phages of the type with which they are lysogenized. They are immune to superinfection. Under some conditions, the prophage initiates synthesis of phage proteins that lead to the destruction of infected cells and release of new phage. This process is called introduction. If ultraviolet radiation has damaged the host DNA, the ensuing prophage introduction is termed UV introduction. When a non-lysogenic F-bacterial cell receives by conjugation a prophage from a lysogenic HFR donor, the recipient cell dies by introduction of the lytic phase cycle. This form of prophage introduction is termed zygotic introduction. Lambda phage lytic pathway, lysogenic pathway. DNA replication, synthesis of capsid, maturation, host cell lysis. Lysogenic pathway, recombination, E. coli chromosome, E. coli. Phage can either both lytic and lysogenic pathways. Phage can enter both lytic and lysogenic pathways. The next step, bacteriophage typing. Bacteriophage typing is based on the fact that surface receptors on phages bind to specific cell surface receptors. Only those bacteriophages that can attach to these surface receptors can infect bacteria and cause lysis. This technique is used for identification of bacteria. And plaque assay. When plated with a lawn of susceptible cells, a bacteriophage will form a circular area of a reduced turbidity called a plaque. Depending on the bacteriophage, when plated with a lawn of susceptible cells, a bacteriophage will form a circular area of reduced turbidity called a plaque. Depending on the bacteriophage, plaque may be clear, such as those formed by phage, or a plaque can be an area containing slow-growing cells. The number of infectious virus particles in a sample can be calculated by a plaque assay. This assay is performed by culturing a dilute sample of viral particles on a plate covered with host cells and then counting the number of local lesions called plaques that develop. A plaque develops on the plate wherever a single virion initially infects a single cell. This virus replicates in this initial host cell and then lyses the cell, releases many progeny virions that infect the neighboring cells on the plate. After a few such cycles of infection, enough cells are lysed to produce a visible plaque on the layer of remaining uninfected cells. A bacteriophage capable of productively infecting a cell is called a plaque forming unit, PFU. Then measuring burst sites. Mutation of either the phage or the bacterial genome or changes in the conditions under which infection occurs can alter the phage-host interaction and the efficiency with which phage replication occurs. Measuring the number of progeny bacteriophage particles produced within and released from an infected cell is a simple way to gauge overall cell expression of the infected phage. The number of mature virus particles released from an infected cell is called the burst size. And eukaryotic viruses, animal viruses. Animal viruses have a variety of shapes, sizes, and nature of genome. The genome of animal virus may be DNA or RNA, or double-stranded or single-stranded, linear or circular, segmented or non-segmented. The general diversity of animal viruses has been classified into 7 groups based on characteristics such as genome type and mRNA synthesis pathway. 6 classes of animal viruses are recognized. Classification of animal virus. Class 1. Genome dsDNA. It is most common type of animal virus. Viral DNA enters the cell nucleus where cellular enzymes transcribe the DNA and process the resulting RNA into viral mRNA. Then class 1A, DNA replicates in host cell nucleus, use host enzymes for viral mRNA synthesis, adenovirus, herpes virus, papovirus, SV40. Class 1B, DNA replicates in host cell cytoplasm using viral examples. And virus minus sense RNA contain a single negative strand of genomic RNA whose sequence is complementary to that of the viral mRNA. The genomic RNA in the virion acts as a template for synthesis of mRNA but does not itself encode proteins. Virus can have several different effects on animal cells. Lytic infection results in the host cell lysis. Persistent infection. In persistent infection, the infected cell remains alive and continue to produce virus indefinitely. Then latent infection. In a latent infection, there is a delay between the infection by the virus and lytic. Wait a minute. Effect of animal virus on animal cells. Virus can have several different effects on animal cells. Lytic infection. Lytic infection results in the host cell lysis. Persistent infection. In persistent infections, the infected cell remain alive and continue to produce virus indefinitely. Then latent infection. In a latent infection, there is a delay between infection by the virus and lytic events. Cold sores caused by the herpes simplex virus are a typical example of latent viral infection. Then transformation. Certain animal viruses can have a normal cell into a tumor cell, a process called transformation. Here you could see attachment and penetration of virus into cell. Tumor cell transformation will be taking place. Lytic infection, cell lysis. Persistent infection, latent infection. So I would like to read this once again. Lytic infection results in the host cell lysis. Persistent infection, the infected cell remains alive and continue to produce virus indefinitely. Latent infection, there is a delay between infection by the virus and lytic events. Cold sores caused by the herpes simplex virus is a typical example of a latent viral infection. Okay. These are possible effects that animal viruses may have on cells they infect. Bacteriophages, viral genomes typically injected with capsid remaining outside cell, no segmented genomes, no 5' capping, no poly A tails. Phage proteins rarely penetrate the host cell. Phage M13 is an exception. The eukaryotic viruses, no tailed forms. No tailed forms. Viral DNA is never injected into host cells. Virion is typically taken up inside cell. Some ssRNA viruses have segmented genomes. 5' end of each ssRNA virion plus strand or viral mRNA is capped. 3' end of ssRNA animal viruses have poly A tail. Viral proteins commonly enter the cell. Retrovirus. Retrovirus are ssRNA plus sense containing animal virus that replicates through a DNA intermediate. Retroviruses are enveloped virus. The enzymes found in the virus particles are reverse transcriptase, invertase, and a protease. The virion also contains specific cellular tRNA molecules. The genome of the retrovirus is unique. It consists of two identical single-stranded RNA molecules. All retroviruses contain the following genes and are arranged in the same order: GAG, encoding structural proteins; POL, encoding reverse transcriptase and integrase; and ENV, encoding envelope proteins. Some complex retroviruses such as HIV-1 contains additional regulatory genes. The family Retroviridae comprise a variety of enveloped RNA viruses such as endogenous retroviruses, leukemia viruses, and HIV-1. The replicative strategy of which includes an essential steps: reverse transcription of the virion RNA into linear double-stranded DNA and subsequent integration of this DNA into the genome of the cell. The virion RNA is 7 to 12 kb long and it is linear, single-stranded, non-segmented, and of positively polarity. Overall process of application of a retrovirus: entrance into the cell via fusion and cell membrane, uncoating of the virion, reverse transcription of one of the two RNA genomes into a single-stranded DNA that is subsequently converted into linear double-stranded DNA by reverse transcriptase and then enters the nucleus. Integration of retroviral DNA into the host genome. Transcription of retroviral DNA leading to the formation of viral mRNAs and viral genomic RNA. Assembly and encapsulation of the genomic RNA into nucleocapsids in the cytoplasm. Budding of enveloped virions at the cytoplasm membrane released from the cell. RNA envelope retrovirus. The viral code is uncoated and it enters the host cell. Viral RNA uses reverse transcriptase to make complementary DNA. Viral RNA degrades. CDN enters the nucleus and is integrated into the host chromosome, forming a provirus. Upon activation, proviral DNA transcribes viral RNA, which is exported to the cytoplasm. In the cytoplasm, the viral RNA is translated. Viral proteins, new capsids and envelopes are assembled. An assembled virus buds from the cell membrane, then retrovirus. HIV and AIDS. The acquired immunodeficiency syndrome AIDS was first recognized in the United States in 1981 in the Los Angeles, New York, and San Francisco when a group of patients displayed pneumocystis carinii pneumonia (PCP) caused by opportunistic fungal pathogen pneumocystis carinii and Kaposi sarcoma, an extremely rare skin tumor. A more complete evaluation of the patient showed that they had a common marked deficiency in cellular immune responses and a significant decrease in the subpopulation of T cells that carry the CD4 marker. The CD4 molecule is 55 Kd protein found predominantly on a subset of T lymphocytes that are responsible for helper or inducer function in the immune system. It is also expressed on the surface of monocytes, macrophages. It is now recognized that AIDS is the first great pandemic of the second half of the 20th century. The disease appears to have begun in Central Africa as early 1950s. Etiologic agent of AIDS is human immunodeficiency virus. It was discovered and characterized by Luc Montagnier and Robert Gallo in year 1983 from the patients suffering from lymphadenopathy. HIV belongs to the family of human retrovirus and the subfamily of lentiviruses. Human retrovirus AIDS is caused primarily by the HIV-1 virus and cases result from the HIV-2 virus infection. This virus is closely related to HTLV-1, the cause of adult T-cell leukemia or disabling progressive neurologic disorder called HTLV-1 associative myelopathy and HTLV-2, which has been isolated from individuals with hairy cell leukemia. Recently, a virus, simian immunodeficiency virus related to HIV-1 and HIV-2, the strains primarily responsible for AIDS, has been isolated from African green monkeys and is believed to be the ancestor of the AIDS virus. Somehow, this virus enters in human and mutated to the two current Africa human viruses SBL and LAV2, which are intermediate viruses. These intermediate viruses may have evolved into the highly virulent HIV-1. HIV, formerly called lymphadenopathy associated virus, human T lymphotrophic viruses, and AIDS associated retroviruses. Structure of HIV-1. HIV-1 has an icosahedron structure. It is an enclosed lentivirus within the family Retroviridae. The envelope develops derived from the host cell. Envelope contains 72 external spikes made up of glycoproteins GP120 external and GP41 transmembrane. These glycoproteins serve as a viral receptor for CD4 on the host cell. Within the envelope is the viral core or nucleocapsid, which includes a layer of protein called P17 and an inner layer of protein called P24. GP stands for glycoprotein and the number refers to the mass of protein in thousands of Dalton. HIV-1 is a retrovirus. It contains 2 copies of plus single-strand DNA genome. Most retroviruses that are capable of replication contain only 3 genes, namely GAG, POL, and ENV. HIV-1 contains 9 different genes in its 9 kb RNA genome. Flanking these genes are the long terminal repeats, which contain regulatory elements involved in gene expression. The major difference between the genome of HIV-1 and HIV-2 is that HIV lacks the VPU gene and has a VPX gene not contained in HIV-1. Function of encoded protein, nucleocapsid protein, enzymes like reverse transcriptase, protease, invertase, ribonuclease, envelope glycoprotein, like that. HIV-1 infects T cells that carry the CD4 antigen on their surface. In addition, this thing we will be learning with the help of next video. Thank you for listening.